Hi to everyone. We have the pleasure today to have Anke to talk about the suspension of E. coli bacteria, the motion of them. And we're really super happy to have you here for so many reasons, but especially for your talk. And Anke is going to also answer all of the questions during the talk, during the class. So you can raise your hand, you can write in the chat. We're going to stop the questions, and we can ask the question during the talk. So no worries for that. And Anke, you can eventually share your
Mhm.
screen, and you can start your talk. So really feel free to ask the question during the talk, and no, don't be shy. It's gonna be fine.
Okay, let me go to the good position.
Yeah.
Okay, so you see the screen well?
Yes. Yes.
So
Perfect.
let me get my, no, not this. No, didn't want to annotate.
Yeah,
Oh,
we
it's
can
here.
see it too.
Yeah, I think I have the
The
laser
laser,
pointer.
fine. Perfect.
Okay, well, thank you very much, Michelle and Leah. I'm really, really happy to be here. And as has been said, if you want to interrupt me, please do so. I think we lost a bit the to be used to talking to our screens. So
Mhm.
I think if you interrupt me and have questions and discussions, I think they will make everything more pleasant. So I would like to talk to you today about work that we've been done over the last years in the group on the transport and the motion of E. coli bacteria. So this is work that has been done by a number of students. So Benjamin and Peixin have just recently defended their PhD. So sorry, I have to move this around a little bit so that I can see my own screen. Norris, Francesca, Martina, and Guan Ying were postdocs and PhD students in the group a bit longer ago. Many of them have now their own positions. And all of this work has been done together with colleagues of myself at the ESPCI, with Eric Clément, Olivia Durour, and Teresa Lopez Leon. And our experimental work has been complemented by theoretical physicists, Andreas Zöttl in Vienna, Arnold Mathissen, who is now in UPenn, and Rodrigo Soko. And before I start, I would like to highlight that you see already a beautiful drawing on the first slide. So these drawings are done by Martina Goral, who was not only a very good PhD student, but can also draw very well. And so you will see her illustrations all along the talk. So as a starter, because I'm not sure how familiar all of you are with active fluids, I want to give a quick introduction. What is a complex fluid or an active fluid? Sorry. So an active fluid, the definition is that it's a suspension of self-propelled particles. Self-propulsion means in this case that the motion results from the dissipation of energy at the scale of a particle. So if you apply a magnetic field from the outside, for example, they would not be called an active particle. And this means that energy is injected locally in the system, and then so these systems are inherently out of equilibrium. And actually, the fact that they are inherently out of equilibrium confers them a number of really intriguing and interesting properties. For example, on these two films, what you will see is that without an external gradient, flows can arise. So the first example are crinkle rollers by the group of Denis Barthelot in what they call a microfluidic racetrack. And you can see that after some competition between the rollers, they decide to go in one direction and you have a flow, a directional flow that is created. And the second example is an example from the Ray Goldstein lab. And what you can see here are bacteria. They're confined in a microfluidic chamber of like 20 microns diameter. And through the interaction with the wall, again, spontaneous flow is created and these bacteria rotate in a certain direction. What is also observed in these active bars is that you can extract mechanical work. So you see these small wheels and these gears, and they start to rotate in a certain direction, something which would be completely forbidden by thermodynamics in just a fluctuating Brownian bars. And finally, what has been observed, predicted and observed, so this is results from an experiment, is that when you add mobile bacteria to a liquid, you can actually decrease the viscosity below the viscosity of the solvent. And in some cases, the viscosity can completely vanish. So you see that there's a number of very intriguing properties, and this is mainly why the scientific community has been very, very interested in these systems. But of course, we also have a number of important applications. So bacteria are everywhere. They're in the soil, they're in the ocean, they're in the clouds. They are also observed by the fabrication of biofuels. For example, if you think about algae, they exist in different forms. They can swim around, they can form biofilms. So for example, in our teeth, we try to avoid these biofilms. And there's many medical applications where these bacteria occur. So for example, we want to avoid upstream contamination in a catheter, for example, or sometimes we want to direct things somewhere and people have developed something like small micro robots that could eventually in the future move through our blood veins and transport things. So it's obvious that it's very interesting and important to be able to control and understand the transport of these microorganisms. But it's also obvious that if you look at the applications, that the understanding is quite difficult due to the complexity of the microorganisms and of the surroundings. So the approach that we took with this situation is the following, that we want to use a model bacteria, so a model microswimmer. In our case, it's an E. coli bacteria. And then we choose specific conditions in which we want to look at these bacteria. And in the case of what I want to present you here, I want to study 3 situations. How do bacteria interact with flows? How do bacteria interact with surfaces? And how do bacteria interact with each other? And the fact that we choose very well-defined model systems will hopefully allow us to gain fundamental understanding. But before going there, I want to introduce you the main actor of the system, so the E. coli bacteria. What you can see here is an E. coli bacteria observed under the microscope, not microphone. So what you can see in green is the bacteria body. You see it wobbles a little bit around. And then in red, you have a bundle of flagella. And these bundle of flagella push the bacteria. And maybe you saw it right now, something has happened. So sometimes the bundle is well formed and then the bacteria swim straight. And what you maybe saw is that one of the flagella left the bundle, then the bacteria tumbled and reoriented. So why does the bacteria have this shape? Why does the bacteria move in this way? It actually comes all back to the fact that bacteria, due to their very small size, live in a world of small Reynolds numbers. So if you are in a world of small Reynolds numbers, everything is dominated by viscosity. And it actually means that if you do just a simple reciprocal movement, then actually this does not lead to swimming. It would just lead you to go back and forth. So this is the first condition or the first thing that bacteria have to work with. And the second is that as there's no inertia, the swimming has to fulfill a zero force and a zero torque condition. So to overcome this, bacteria have actually developed a specific strategy. And for the E. coli bacteria, they break symmetry due to the chirality of their flagella. And so this is how a typical E. coli looks like. It has the head and the flagella. So this flagella will actually rotate, so they're rigid. They will rotate at a certain frequency. And as we have to fulfill a zero torque condition, so not only a zero force condition, when the flagella rotate in one direction, the head has to rotate in the other direction. And I just want to make a small interlude because I think now we know the system very well. So we actually believe that there's rigid flagella and that they actually rotate. But when this was first suggested, people could not believe that this is possible. But because what that means is that you need a motor. Where the flagella are connected to the bacteria body, there must be a motor that rotates the flagella. And actually, indeed, this is true. This motor exists and has later been observed. So this is from a paper by Berg, who's one of the pioneers working on E. coli bacteria. So this is actually an electron microscope image. And what you can see here is 45 nanometers in width. But if you look carefully, this is a very classical stator and rotor motor. So you have the stator, you have the rotor, and actually you have the hook, and the thing will turn. And it's a small miracle, I would say, of nature. And actually, it's a proton gradient. So the proton gradient brings the energy, but mechanically, it's just a stator and motor rotor. And so this is what drives the flagella rotation, which then allows the bacteria to propulse. But there's another thing that is possible. The motor can either turn in one direction or it can switch and rotate in another direction. And this turns out to be extremely important. And you have already seen this on the film. So when all the flagella rotate in the same direction, in a specific direction, hydrodynamic interactions bundle the flagella and they form like one big helix and they push the bacteria forward. This is called a run. Bacteria go straight. And then from time to time, one flagella will change direction. And this is actually what is depicted here. And when the flagella changes direction, the hydrodynamic interactions do not bundle it with the rest of the flagella anymore. It leaves the bundle, and then the bacteria tumbles around, changes orientation, until at some point the motor switches back, the flagella goes back into the bundle, and the bacteria continues straight. So that's a very elementary way of exploring space. It's called a run and tumble mechanism. So when the bacteria is very happy, it will tumble a lot because it will remain more or less static where it is. And when the bacteria looks for food, it will actually have long directions of runs to explore space in a large way. And so we can actually look at this and look at a 3D trajectory of a bacteria. And I will tell you in a second how we can actually record those. And what you can see is that a happy bacteria without any chemical gradients or other stimuli will perform something that looks a little bit like a 3D random walk, triggered by this run and tumble motion. So if we summarize, so what we have seen about these E. coli bacteria, so they have very specific properties. First of all, they have a specific way of exploring the space. They have a certain swimming velocity, and they have this run and tumble motion. Then also there's noise in the system. Bacteria are small, they're on a micrometric scale, so they have like rotational noise from Brownian fluctuations, but we can also see the run and tumble motion as some kind of noise. And then you've seen they have a quite complex shape. Overall, they're elongated, they look a little bit like a fiber, but in reality, they have a head, they have a flagella. And also, which I have not said explicitly yet, when you put bacteria in a surrounding fluid, they will create a disturbance flow. And so this disturbance flow comes from the zero force and zero torque condition. Zero force condition means that actually if the flagella pushes in one direction, the head resists in the other, and they create something which is called a dipolar flow. So the bacteria will like push liquid outside along their axis and pull it back in from the sides. And then you have higher order terms which come from the fact that the head and the tail rotate in different directions. So now that we know how individual bacteria look like and what their properties, we can now wonder how do these properties influence the bacteria when they're transported, when they have a surface close to them, or when more of these bacteria are close to each other. So let me start with the first example. Let's put bacteria under flow. So what we do a lot in the group is microfluidics. So microfluidics has the advantage that we can very nicely control flow conditions, and it's actually also very easy to observe. So the first experiment I wanted to show you is a very simple setup. We put like one E. coli bacteria, something in a very dilute suspension, in a pozoid profile. So our channel is like 100 micrometers high, it's 600 meters wide. It's like a Halo short channel. And what you see here is actually the pozoid profile that will develop in the height of the channel. And then we will put bacteria in there and see what happens. So let's first think about conceptually what we expect. So we know that if we put particles in a flow, there's fluid structure interactions between particles in the flow. And if I have a passive object, like a passive fiber, then it would just tumble around. But while the fiber is tumbling, it would just be transported along the streamline. However, if this fiber is active, orientation becomes crucial because actually when the fiber swims, if the fiber is oriented in a certain direction, it will also swim in this direction. So basically what happens if I put an active object in the flow, the swimming trajectory will be fully given by the orientation dynamics. And if I look at average velocities, it will be the orientation distribution, maybe in presence of noise, which will actually give me where bacteria will swim. So that's a very interesting problem to look at, but technically now you have to be able to follow a bacteria, which you don't know what direction it will go, in an external flow. So together with Thierry Darnige, which is an engineer in our group, we have developed a Lagrangian tracking, which is something which allows us, by displacing the microscope stage, to follow a bacteria in the 3D space. So basically what you do is you have an image, you analyze your image, then you displace the stage in different directions. It's very easy, well, very easy. It's easy or straightforward in X and Y. So you analyze your image, and what you do is you keep the target bacteria in the center of the image frame. It is actually more difficult in the Z direction because in Z you have to find out if your image is in focus or not. But if you just look at one image, you don't know if you're above or below the focal plane. So typically you need two images to compare to know if you move in the good direction. Now recently, Thierry and Eric have made a lot of efforts to implement AI image treatment techniques. And now very often the system can already recognize in one direction it has to go. And the tracking has even been improved a lot compared to that. And so let me just show you an example. I also want to mention that we here, what you see, the dot is only the body of the bacteria. Because if you have a bacteria body and the flagella, it's actually a very elongated object. So depending on the orientation, its shape will change a lot on the image. So that's difficult for the image treatment. But if you only take the body, then actually it's nearly spherical, not exactly, but so the differences will be small. So what you can see here is we follow this bacteria. So here you see that the bacteria is close to a glass slide. You see that close to the glass slide, the bacteria swims in a circle. What you can see that soon the bacteria will throw itself into the bulk. And you could see here also there were dead bacteria stuck on the glass slide. These bacteria now have vanished. We still follow the bright dot, and you will see that when it comes close to the bottom glass slide, again, there's some dead bacteria stuck on the bottom glass slide. So you see now we reach the bottom, but still we can follow the bacteria quite nicely. So this was a system that already worked very well, but we were a little bit frustrated because we couldn't see the flagella at the same time. So together with colleagues from Edinburgh University, we developed a two-color system to track bacteria, where now the body is green and the flagella are red. So we have a beam splitter, we track only with the body, but once we are done, we can put the flagella back. And this is the nice film that you saw in the beginning. So now we can follow bacteria on their own trajectory and transport it in flow. And at the same time, we have the full information of the configuration of the flagella. So now somehow we are ready and we can start to do our first observations. So this is one of the first observations now that we did with the trajectory on an E. coli bacteria in this helixal cell. What you can see is the bacteria started from the bottom surface, and actually it does this surprising going back and forth 8 loop through the geometry. It actually goes towards the middle, then it bends back, it reaches nearly the other surface, and it does this several times. So how can we explain this movement? So let's go back to the basics. So it is well known what a rigid rod does when I put it in a shear flow. So these are actually called Jeffrey orbits. So a Jeffrey orbit describes the dynamics of an elongated object in a simple shear. And so if you just think that you're in the shear plane, what would happen, it will actually flip around with the flow. It will, my screen doesn't really want to show this fiber, but, and once it's aligned with the flow, it's a more or less stable situation, and then it will flip again. So it spends little time in the orbit and more time aligned. But the full Jeffrey equations are a little bit more complicated because you can have different types of tumbling motion depending really on the orientation of your fiber in the beginning. But these are very classic equations. And so basically the dynamics are described as the dynamics of two angles, so the in-plane and the out-of-plane angle. And the only parameter of the system is the aspect ratio. So if you want to apply like Jeffrey orbits to bacteria, what we can do, we take these Jeffrey equations, and the only thing we do is we have an effective aspect ratio and we add a swimming velocity on top. So this was the most simple approach to see what would happen. And I just want to show you a quick simulation, which will also be important for the following. So to simulate these Jeffrey orbits, what you imagine is that you have this fiber that actually the tip of the fiber goes around on a unit sphere. And depending on where you start, you will go through different trajectories. And this is actually the unit sphere is folded up here. So if you start on a certain orbit, you will stay for the rest of your life without noise on this orbit. And so that means if your head is on the right side, it will always stay on the right side. So let's see if this very simplified model can explain what you observe for bacteria under Poiseuille flow. And so indeed, it actually makes sense because the Poiseuille flow is a very specific flow in the sense that the rotation rate given by the shear rate actually changes sign at the midplane. So if you think about the rotation rate given by the shear, the rotation component, if I start at the bottom, I will be rotated around in this direction. Then when I cross the midplane, so actually rotation is weak because I have no shear in the midplane. But once I reach the other side, I will actually bend towards the other direction. So just putting together the rotation dynamic of a slender object and adding velocity on top explains quite nicely qualitatively the observations. But we can go a bit further. And so these are several trajectories that we recorded. So we made our life a little bit easier in the sense that we only recorded trajectories that were so kind to really swim in the shear plane. Right? So they were all oriented in the shear plane. And so you see these swinging trajectories that I showed you already. you can also see sometimes if the shear is very strong, bacteria are rotated so quickly that they go a little bit up and down, but they don't manage to change sides in the Poiseuille flow. But also some of them that actually come from the bottom surface, if they leave towards the flow, they will be just bent back very quickly, or they just go from one side to the other. And these experimental trajectories can be very easily reproduced in numerical simulations with exactly the same ingredients. And that's something that had been predicted by Zöttl et al. A long time ago in baptized active Jeffrey orbits. So this actually works quite nicely with a very simple model. But then we also wanted to see if now I'm not perfectly in the shear plane, but I'm a little bit out of plane, what would happen. And this is from a numerical simulation. And what you see is that when the head is a little bit on the side, basically you have this kayaking motion, but while you kayak, you also swim towards the side. Okay? So if you look from above, it means that you drift towards the side because your head looks a little bit towards the side. But while doing this, you will actually orbit around. So we can also observe something like this in a projection from the experiment. But now we could still wonder, does this really work for long times? Because what you see from the experiments is like 1 or 2 loops. So we would first of all expect that noise sets in at some point. And also in reality, the bacteria are actually much more complicated than just an elongated shape. So to go a bit further, we wanted to really look at the full shape of the bacteria. And so we were not the first to look at that. So there's something that was baptized Rheotaxis in Mattia, and it's something that has been observed by the group of Roman Stocker. So what he saw that if you put many bacteria under shear flows, then you actually see that they seem to start to drift in the vorticity direction. So they seem to orient perpendicular to the flow direction. And this has been explained by a purely, again, fluid structure interaction effect. So it has been shown that a chiral object, so a helix, if you put a helix in a shear flow, this helix will actually drift into the vorticity direction. So that's really a slender body interaction with the fluid, which if later somebody's interested, I can explain in more detail, and actually leads to a displacement of the helix in the vorticity direction. However, the head, you can imagine the head like an anchor. So the head is much more solid and has much more viscous friction. So basically the head is held fixed. The helix wants to go on the side. And as they are coupled, this will actually lead towards a torque. So the shape of the bacteria induces what we call the rheotactic torque. And this rheotactic torque wants to orient bacteria absolutely perpendicular to the flow direction. So we wanted to look in detail how this looks like and how does this intervene. So now to do this observation, we didn't just look at one Jeffrey orbit, but we looked at transport of bacteria over long times. And for this, we looked at statistics. And so it's the same setup, but now we are not following trajectories, but we're looking at different planes in our Poiseuille flow that you can see here. So different planes, different distances from the bottom means different shear rates. And we just wanted to see what are the average directions and flow speeds of the bacteria. So already let me show you a general observation. So these are bacteria trajectories in a 2D plane, close to the bottom and close to the top. And I hope you can see that for this one, bacteria drift upwards, and for this, they drift downwards. So if you now imagine that the flow goes from left to right, those at the bottom see a different sign in the shear rate than those at the top. So we can indeed imagine that this is this rheotactic effect, where bacteria are oriented towards the vorticity direction, which would be perpendicular to the flow, and apparently don't go downstream in a straight direction. So to characterize this, what we have measured is what we call the rheotactic velocity. So only the component of the velocity which is perpendicular to the flow direction. Okay? So when we do this, we get something like this. So what you can see is different flow rates. So 5, 10, 20 nanoliters. You can see that this velocity, the drift velocity, increases with flow rate. Makes sense. It is 0 in the middle, also makes sense because there's no shear rate in the middle. It changes sign when you go through the middle. And of course, it's also stronger when you're close to the surface, because close to the surface in the Poiseuille flow, the shear rate is highest. I also want to point out that we did not only measure bacteria, but we also measured passive traces. So passive traces have no reason to deviate from the streamline, and luckily they don't. So you can actually see that for the different flow rates, they all have a 0 drift velocity. So this turned out to be a very important check and alignment test of our setup, because we later on really wanted to measure distributions of bacteria velocities, distributions of orientation. So we had to make absolutely sure that we were perfectly aligned with the flow direction. So now these results, one has the impression that the control parameter is the shear rate. So what we indeed did, that we plotted this drift velocity as a function of the shear rate. That's what you can see here. And we actually obtained the same shear rate in many different ways. We changed the flow rate, we changed the position in the channel, and all of the data actually overlaps if we do that. And also, I should say there's simulations on top of that I will introduce a little bit later, and all the experimental and simulation data actually collapse. But there's a number of questions that arise if I look at this graph. First of all, we have normalized this drift velocity by the average swimming velocity of the bacteria. So I told you in the beginning that bacteria want to orient perpendicular with the flow. So if bacteria orient perpendicular with the flow, then the drift velocity should be equal to the swimming velocity. But we see that we stabilize or kind of stabilize on something which is 0.6, which is quite far from 1. So apparently bacteria do not perfectly orient perpendicular to the flow. And also this drift velocity increases with the shear rate, which somehow doesn't disturb us. But on the other hand, what determines the slope? Right now, we don't have a different time scale in the system. We just know that the rheotactic drift wants to orient bacteria perpendicular to the flow. So it's not obvious why they should depend on the shear rate. So we wanted to understand these two observations by looking into more detail in the system. So our system allows us not only to measure average velocities, as you can see here, but we can also measure velocity distributions. And so we measured the velocity distributions of bacteria, and they are represented here as a function of as a function of the shear rate. So basically from here to there, we increase the shear rate. So what you can see, if there's no external shear, bacteria orientations are random, which is the black line here. And then when we start to increase the shear, we see that slowly bacteria velocities go more and more towards the right. I just say right for simplicity. I mean, they orient in the vorticity direction. But what you can see that the peak in velocity, it remains really broad, even if the highest shear rates that we have looked at. So that's already an information, but we also know that the velocity is only a consequence of the orientation dynamics. So we also measured bacteria orientation. And now if you look at that, again, you can see no shear, nothing happens. But if you ramp up the shear, bacteria will orient towards the side. But what you can see is that there's not a single peak. So bacteria are a little bit above and a little bit below the perpendicular orientation. Okay, that's what you see here. Perpendicular would be at p half. So this double peak, we cannot see it in the velocity, because if I project from here to there or from here to there, is the same thing. But it gives us already the idea that something a little bit more complicated in the system. So now with this, we would like to understand where this comes from. So we go back to the theory and to the modeling. So apparently the Jeffrey is not enough. So we know there's this rheotactic drift, and Andreas and Arnold have managed to cast this rheotactic drift in a simple form that we could just add to the Jeffrey equation. So again, we just added to the dynamics of the angles. And what you can see again is that this rheotactic drift is also proportional to the shear rate, to gamma dot. We have a new parameter, however, that arises. This new is the rheotactic strength. And also what I should tell you, this rheotactic strength is actually 10 times weaker than the Jeffrey term. So this explains, if I just look at 1 or 2 loops of a Jeffrey trajectory, I don't really see a difference. But if I look at distributions over a long time, I really have to take this into account. And so these are the new equations that can describe the dynamics. And then we can also, of course, add orientation and noise on this. So let me just quickly go back. So we had seen, So we had seen that the simple Jeffrey orbit head on one side will stay forever on one side. Now, if I go to the situation where I add the rheotactic torque, you can actually see that if I start with my head on the wrong side, then actually you see here that I will loop outwards. So the amplitude will get bigger and bigger of my fiber or filament or whatever, until it actually stretches over the other side. And now it has changed side, and it will slowly go towards the perpendicular direction, which you can see here, to end up on the good side. So this is deterministic, so there's no noise yet, but we can already see that the dynamics is very different if I add a little bit of the rheotactic torque. Okay, so now there's a preferred orientation with the head on a certain side. But to understand this better, we also had to add the orientation and noise. And so this slide is a little bit full, so you have to bear with me. So what is shown here is, again, these orbits of the Jeffrey dynamics. And we have a situation where I have no chirality, and I have a situation where I have chirality. So you recognize these Jeffrey orbits where once I'm on an orbit, it's closed, I will stay forever. And you can have here these trajectories where I can actually change sides. And then, so we did a second thing. So from here to there, we actually ramp up the shear rate. So that means that the bottom situation has a lot of noise, and on the top of the situation, the shear rate starts to dominate more and more. So in a system with a lot of noise, actually, I have a, well, not exactly random, but a close to random orientation. But I see already that there's a preferred direction of the fiber upstream or downstream. So Jeffrey does not distinguish between head and tail. So if I only have a Jeffrey equation, being oriented upstream or downstream is the same thing. And then I start to ramp up the shear rate. So if I don't have chirality, it just means that I become more and more oriented downstream or upstream. However, we see if I add the chirality terms or the rheotactic drift, it dramatically changes behavior already at quite small shear rates and actually bring things towards the right side. And we can also recognize that these two peaks that we saw in the distribution, they're actually reminiscent from this upstream and downstream orientation. And they're just slightly pushed together when I go towards the side. So that's what you can see here. So these two start to merge together, get closer together, and actually merge towards the perpendicular orientation. But to analyze this further, now we can actually analyze the system around these specific points. So first of all, we can look at this fixed point, and actually turns out that this is a fixed point, perpendicular, but it's only marginally stable. So that means that things take an infinity of time to really reach the perpendicular orientation, and they will take a long time to spiral towards it. And the second thing we can do is then we can take these two preferred orientations without the rheotactic drift. And here what you can actually see, we can show that they are linearly pushed together by the rheotactic drift against the noise, and this explains the linear increase. So with this, we can fully explain the rheotactic curve that we observed. The linear slope in the beginning is actually a competition between the rheotactic torque and rotational diffusion. And the fact that we stabilize only so slowly on the perpendicular orientation comes from the fact that it's only marginally stable. So with this, we have fully understood this mechanism. However, I want to do a second small interlude. So bacteria are a great system in the sense that if you want to have many of them, you just grow many of them. They're very reproducible, but there's nothing that you can do to change the geometry if you would want to. So and we think that the rheotactic orientation really comes only or mainly from the geometry of the system. So we thought maybe we can produce something which is a passive bacteria. And so I'm always very happy to say that we do passive bacteria because many people do active colloids, but we do passive bacteria. And so we have a very nice device at the ESPCI, which is called a nanoscribe. And so this nanoscribe allows to 3D print inside a resin very, very tiny objects. And so in this way, we could actually produce, together with Justine Laurent, which is an engineer, who is an engineer at the ESPCI, these beautiful small, bacteria-like objects, where the head is like 10 microns and the flagella are 30 microns. So they're a little bit bigger than the real bacteria, but they're actually still very close. And the advantage is also that they don't have any Brownian noise. And the nice thing is now I can actually tune the helix exactly as I want to, and I can even knock out the chirality by printing a double-sided helix. And the way we do the experiment is that you print the small helix, and what you can see here is that you get close to the helix with a capillary tube. You actually detach the helix from the wafer that it was printed on, and then you suck the helix towards the capillary tube. And now with this tube, you can bring it into a bath, and then you can release it, and then you can flow it in a channel and look at the dynamics. And I will just show you these two films. So these are two of these passive bacteria under shear flow. So they're exactly the same, but we changed chirality. And so you can really nicely see these Jeffrey dynamics. There's the fast oscillation method, and there's a slow reorientation. And if we wait long enough, indeed, they will reorient perpendicular to the flow direction. but with the helices in opposite directions. So I won't have time to tell you more about that, but it actually proves that the effect really comes from a geometrical effect. Okay, so this was a lot on bacteria and the flow. I want to talk a little bit about bacteria at surfaces, and I hope to keep time to talk about the collective motion. But maybe if somebody wants to say something or ask a question, it could be a good moment.
Yeah, you can ask the question.
Yeah, hello, and thank you in advance for the great talk. Just a question regarding this 3D microprinted fake bacteria. Could it be possible then to compare with a population with high concentration of real bacteria? Could you compare to see if there is an effect, for example, of biofilms or something like this? Like compare if there is some effect of the fact that it's bio or not on the behavior.
Yeah. So somehow is what we wanted to test is actually maybe we wanted to decouple the bio effect from the geometry effect that we can control. I think the most differences that we see is that there's noise in the biological system. So biofilm is something we normally avoid. So the way we treat our bacteria is that they are happy, they have enough to eat, but not so much that they multiply, and so they don't form biofilms. But indeed, the idea was to take out one element of the bacteria, which is the shape, and to see what kind of behavior we get with the shape. And somehow the model allows us to compare quantitatively if in the bacteria distribution we have all the information we need. Yeah. I have also to say what turned out to be a little bit different here is that actually in this case the head was a bit more heavy than the tail, because when you 3D print, you print several times over the head and only once over the helix. So also we created an object that was head heavy, because you saw here they actually stabilize quite fast on the perpendicular direction. And this is because gravity helps them to stabilize.
I see. Okay.
Yeah.
Thank you very much.
Yeah, but there's many things that could be fun to try with these systems.
Thank you.
There's something else? No, not that.
No.
I can move on.
Well, just a very quick question. You just mentioned gravity. What's the direction of gravity here?
In the plane.
In the plane.
Yeah.
Okay.
So actually, those are in general a little bit more heavy. So the microprinted ones are close to the bottom surface, and we observe from below.
Okay.
So gravity is either like this or like that. And so for them, it plays a role. And the fact that the head is a little bit more heavy, I mean, you see they still can do these big oscillations, but it actually brings them down a little bit more every time. And so they get to this rolling state faster. And for bacteria, as they swim and they have more tiny, gravity actually doesn't play a role. And we also density match the bacteria with the surrounding liquid. Yeah.
Okay, thanks.
Okay. Good. So it's a bit artificial somehow to look at bacteria inside the bulk, because bacteria actually like to be close to surfaces. So I will just have some slides on the surface to tell you what happens close to surfaces. First of all, if you look at the dynamics of trajectories of individual bacteria close to surfaces, you see that they're even more complex than if you look at them in bulk. And depending on the shear rate that you apply, you can go from swimming in circles to upstream swimming, to being bent downstream, to have a bit of rheotaxis to the left and to the right with oscillatory trajectories. So that would also be a whole talk in itself. So I will not tell you everything about it, but I want to concentrate on this fact that actually bacteria can swim against the flow. This is also somehow a purely geometric effect. You have the head. If you imagine I'm a bacteria, I'm close to surface, I have the head acts like an anchor, and now I apply flow. So basically what will happen is that the flow will rotate the tail behind the head of the bacteria. It's like when you have your church tower, and on top of the church tower you have these flags or the cock or whatever, something that tells you the direction of the wind. It's something where actually it pushes the flag behind you against the wind. So for the bacteria, this means that the flow rotates the helix behind the head so that the head will face upstream. So it's again a purely geometric effect that if the flow rate is not too strong, putting a little bit of flow will actually somehow do the opposite of what you think. It will not flow the bacteria away, but it will actually bring the bacteria to swim upstream. So once we had observed that, we thought, wow, I mean, this is something which creates upstream contamination. In many cases is absolutely not what you want to have. So let's maybe investigate this in more detail. So what you see here is we have a reservoir with bacteria, and we have an inlet where we actually inject clean solution into the system. And then we wanted to see what happens in the channel in between the two. So what you can see here is at the very beginning, the dots again are bacteria. So then we have the reservoir and we scan through the channel. And at the beginning of the experiment, nobody has reached the channel. A bit later, there's more bacteria in the reservoir. And when we start to scan through the channel, you see that actually some bacteria have ventured up the channel. And if you wait even longer, you will see that there's even more bacteria that have actually swam against the flow in the small channel. And it's maybe easier to see on these snapshots that what we can see is that after some minutes, some bacteria have traveled over 13 millimeters. So after some minutes, some bacteria have actually covered microscopic distances. So how can bacteria be so persistent to swim against the flow, and how can they, well, what is the distribution of what happens here for this upstream contamination? So a straightforward thing to do is to measure the concentration profiles in these channels. So if you measure these concentration profiles, here's what you see. So if you wait longer, what you can see is that the bacteria concentration in this part in the reservoir increases. But mainly also what you can see is that this front of bacteria that penetrate into the channel actually gets, well, larger and larger, and also further and further inside the channel. And something which is quite clear also is that we have these very long tails. So there's some bacteria which are extremely persistent and swim very, very, very far in a short time. So main aim we had here is to understand why some bacteria are so persistent and why are there so long tails of things contaminating the channel upstream. And so to do this, actually I have to try to show this film to you because it doesn't really want to work in the way I think it should. Let's see if I can make this work. Okay, let me take the razor. Okay, so, oh no, okay, nothing. Okay, I will tell you what happens here. I don't know why I have a problem with the format of this film. So this film should tell you that many bacteria swim upstream along the edges of the channel because the edge is a very protected place in the channel. So they go to the edge, they swim upstream, and from time to time, a bacteria detaches from the edge. So what you should have seen is that they detach. They are inside the channel, and once they're in the channel, they actually transport it downstream with the bulk flow. So we can make a minimal model to see how far bacteria can go, by see how far do they swim upstream, and then they transport it downstream, and then they swim upstream again. And the main ingredient that we have to, oh, there are some trajectories. And the main ingredient that we have to understand is how and why do bacteria detach from the surface. So there was a hypothesis that actually a bacteria detaches from the surface when the bacteria opens the bundle for a tumble, because that makes the bacteria very, well, fragile. And so to look at this in a bit more detail, we actually used the 3D tracking technique that we have that can tell us trajectories of bacteria together with the exact shape of the bundle. And we analyzed, well, bundling and unbundling phases, which you saw a very nice unbundling here, close to surfaces. So here we have a 3D track of a bacteria. Now it's without flow. So the first situation is a bit simpler. So we look at the trajectory of the bacteria close to a boundary, and everything that is colored in red is when we observed visually the bundle to open. And you see the example down here. So the first observation you make is that there's a lot of tumbles that do not necessarily all lead to detachment. However, when we looked at the statistics, so every time there was a detachment, there has been a tumble before. So the main observation is to detach, I need a tumble, but not every tumble leads to detachment. So that was the first information. And then the second information, we quantitatively measured the residence time of bacteria at surfaces. And what you can see here is this is the distribution of residence times. And again, you see this very long tail. So some bacteria seem to stay very, very long time at the surfaces. So in reality, this is actually a log normal distribution, if we plot it as a log normal, which is characteristic of distributions with very long tails. So to understand this in more detail, we looked at what happens in the bulk, and we looked at the run and tumble distributions. Because to understand how long I stay at the surface, basically I have to understand the frequency of tumbling. And so it actually turns out that the frequency of tumbling also has this very, very wide distribution, is also a log normal distribution. So it means the way that bacteria tumble, so the run times, so the time between tumbles, is a distribution with a very long tail. And from what we know in the bulk, we can translate this to the surface. And this explains why we have these very long tails and why some bacteria are very persistent and stay a very long time at the surface. But we can do one step further. We can now wonder, okay, we know sometimes a tumble leads to detachment, sometimes it doesn't lead to detachment. And actually, what we can imagine is a very, very simple model. Detachment reorients randomly. So if I point outwards, I will leave. If I point inwards, I don't leave. But it also turns out if I leave with a very small angle, the chance that I'm captured again is too high. So it turns out that actually the probability to leave is 1/3. So in average, I need 3 tumbles to leave. And there's one additional information that I can find here. So I told you that this runtime distribution is very large. And this comes from the fact that bacteria sometimes are in a mood where they tumble a lot, and sometimes they're in a mood where they tumble very little. And so what is colored here is the mood of the bacteria. So when they tumble very little, they follow perfectly this distribution of I need 3 tumbles to leave. However, these bright colors are bacteria that tumble a lot. So if I tumble all the time, I'm not efficient either, because I try to leave and then I tumble again and I'm reattached again. So from this, we can learn that, first of all, the residence time is given by the frequency of tumble. I typically need 3 tumbles to leave, but there's 2 populations that stay long at the surface. Those that, well, tumble very little, so they stay for a long time, and those that tumble all the time because they will be reattached. And we think this is the key to understanding what happens for the upstream contamination. But with this, we can also do a bit more fun things. We can now try to construct geometries that allows us to use this. And this is a very recent experiment where we actually have a funnel geometry. And so you should not misunderstand what happens here. For those who do granular media, this is a hopper, but actually the flow comes from left to right. Right? So the flow comes from left to right, but the bacteria accumulate downstream of the hopper and not upstream of the hopper. And we explain this by the fact that they swim along the walls for a very long time, and they actually detached when they reach the constriction because the flow rate is very high there. So it is a hopper flow, but the flow doesn't come in the flow direction, but the flow comes opposite of the flow direction. So there's many more things to be done around that and to play with the system and the transport against the flow along walls and edges. Do I have a bit of time for the last, or you want to ask maybe some questions here?
You have certainly time to talk about the last part, but if there is any question, you can go ahead.
I do have a couple of questions about this. Maybe you said it and I missed it. I don't know. If yes, I'm sorry. But
no worries.
Why do they tumble?
Okay. So I did not really explain why. So basically, the reason they tumble is the way that allows them to change direction. So it's the run and tumble is the basic mechanism the bacteria explore space. So they're not very intelligent organisms. They cannot decide. Yeah. Okay.
So is it a random or?
Okay. Yeah. So it's a biochemical process, and it's not random. It depends on some stimuli, and it depends on some levels of protein. So it's a quite complicated process, but it's absolutely not random. So if the bacteria, for example, are in good surroundings all the time, already in this case, they can be in a situation where they tumble a lot, and they can be in a situation where they tumble much less. So basically, it's a 2-well system, and you have to jump from one well to the other well to go from one direction to the other direction. But the barrier between the 2 wells can change height as a function of things. Stimuli, yeah.
But can you control it? Because you showed that they can have different moods. So can you
Yeah.
tune the mood or?
So the different moods depend on the concentration of a protein.
Okay.
So you could influence this by giving different concentrations of this protein. It's also influenced when bacteria do chemotaxis or follow certain external stimuli. And if you don't specifically control it, it will actually vary and fluctuate with a very long time scale. And this time scale will lead to bacteria that sometimes tumble more and sometimes tumble less.
And keeping on with this tumbling motion, so you show that when they tumble, you usually need 3 tumbles to detach from the wall, from the surface, and when they do, they are just transported back into the reservoir practically. Do they eventually reach the other surface, or they always just get?
No, it's a very good point. So yes, they can also reach the other surface. And maybe here, what you could see here, here you see these 8-like things. This one, for example. So here you see this, that some actually go from one edge to the other edge. So yes, absolutely.
Okay, so
They reach,
the geometry also has an effect on the
Yeah,
concentration along the.
absolutely.
Okay,
Absolutely.
perfect.
Yeah,
I mean,
yeah.
and when I say geometry, I really mean just the width. So the
Yeah,
distance.
exactly. Also
Okay.
the height. They're transported downstream over a certain length, which corresponds to the time they need to reach another surface. Yeah,
Okay.
absolutely.
Great. Thanks.
Okay. Still have energy for the last part. It's the most recent part, so okay. So we talked about bacterial flows, bacterial surfaces, but of course, in many situations, bacteria are not alone. And actually, but the disturbance flow that bacteria create around themselves is actually very long range. So when one bacteria has another bacteria, even relatively far away, they will interact with each other. And what actually happens is that these disturbance flow, I told you, it's like this pusher-like flow. It's flow that goes out along the axis of the bacteria and it's aspired on the side. Actually leads to the fact that if another bacteria, which is an elongated object, comes close and will align. So these long-range hydrodynamic interactions have been shown by kinetic theory to predict a long-wave instability, meaning alignment of bacteria in the same direction. And indeed, experimental observations show these large-scale collective motions. So this is a bacteria bath, and you should keep in mind the scale bar here is 100 micrometers. Let me see if I can play this again. And so the structures that form are several hundreds of micrometers, so much, much, much larger than the size of a bacteria. But one question was not really resolved is there an intrinsic length scale of the patterns observed? So there were contradictory observations from experiments, and some theories implemented a mesoscopic length scale. So this was the question that we wanted to address. And so to do this, actually we built an experimental setup where we wanted to vary the confinement height from very confined to very unconfined, from 20 to 2000 microns, and also vary the bacteria concentrations very widely, from 0.1 to 10%. And to make all of this work, actually in this setup, all surfaces are transparent to oxygen to keep bacteria alive, even in high pools and for high concentrations. And then the idea is to have these bacterial baths, but what we track are not bacteria, but tracer beads, and this is the trajectories you can see here. So from this, we wanted to investigate the properties of these collective motions. And the first thing is that you actually have to define an onset of collective motion. So what you can see here is at a fixed height, you see the trajectories of these tracer beads in the bacteria suspension. And visually, you have the impression that maybe here I start to have collective organized motion. That is not something which is extremely easy to define just visually. So we thought it would be nice to have a quantitative criterion for this onset. And actually, we looked at the velocity correlation function. So velocity correlation function means I choose one point and I look at the velocity at this point over time. And then I get a correlation function, I have a typical time scale of this correlation function. And it turns out that if I wrap the up through different concentrations, that around a critical concentration, this time scale diverges or gets very large. And that is something which is a bit reminiscent of a second order phase transition, where actually close to the transition, we have what is called a critical slowing down of the dynamics. And actually, in analogy with that, and following some theoretical work, we decided that our criterion for the onset of collective motion is the maximum of this time scale. And so by doing this, actually, this is a huge phase space of work that Benjamin has looked at. So he did many experiments with different concentrations and different sample heights. And what you can see is all the points investigated. And actually the maximum in the correlation time are these blue points here. So this is concentration and this is height. So what we can see that is a quite good approximation, the critical density for the onset of instability is inversely proportional to the height. So the more confined, the higher the density of the suspension has to be. And this is in quite good agreement with kinetic theory. However, there's something which is a bit surprising, is that actually in our case, we never observe a saturation. So we know that in our system, we also have noise. And actually noise somehow should act against the collective motion. So if I have a very dilute system, at some stage it should be so dilute that the noise gets so strong that the material do not manage to align again. And this has also been predicted by theory, that at some stage, these onset concentration should actually plateau. But if we look at what is predicted by the theory as a function of the noise, we would expect the plateau value to be much higher. So from these observations, we now have a very nice criterion that can tell us from our experiments where is the onset of instability. If I want to study any properties of the patterns that form, I know exactly how far away I am from instability. I have something which works well with what has been predicted, but there's still a little bit of things that are not fully understood. So what is really the noise in this system? So there remains much more to be understood. But the main thing we wanted to find out is about the wavelengths. So now we can indeed look at the wavelengths. We look at different heights. We are always at the same distance from the onset of instability. And here we have actually scaled the patterns that what you see is always 3 times the height. And you can see that more or less on all of these images, the length scale looks the same, which would mean that the length scale scales exactly with the height of the system. And so again, to measure this quantitatively, we look at correlation functions. So now we look at correlation functions as a function of distance. And what you can see is that you have this very strong decorrelation and things get negative. So when the correlation function gets negative, it's actually the signature of a vortex. Okay, so you have a counter-rotating vortex. So there's 2 scales possible. So either when it goes to 0, which is something like the vortex size, or we just defined a half height of the decay function. And if you plot those, this is what you get. So it seems that for the highest confinement height that we could go to, everything just scales linearly with the confinement. So you see that from a height of 20 microns to a height of 2000 micrometers, the length of the vortices that we observe are always identical to the height. So this already gives a strong indication that there's apparently no intrinsic length scale in the system, so that the system is actually length scale free, and it just scales on the only length that the system gives him, which is in this case the height. But from this actually, well, I should go slowly because this was a surprise. But from this actually, we made even a different observation, because somehow, well, it's great. It's 2000 microns is already quite a lot in the height, but somehow you want to know, isn't there somewhere a length where things break down? And so actually there's another scale in the system. So there's the height of the pool, but there's also the radius of the pool. And actually one day, Benjamin made this amazing observation that when he came to his sample, he found one big rotating vortex. And so now this is 1 millimeter. So this is several millimeters in width. And this vortex was so stable in time because the observation window on the microscope is only one of these small windows. And it could move around and actually observe different parts of the sample and stuck together the vortex afterwards. So it seems that I can have even larger vortices that scale on the other scale lengths in my system, which is the radius. And this actually has not been observed as regularly as the turbulent motion you saw before, but still is something that can be easily obtained. And so again, in this case, the wavelengths of what is observed here is linearly given with the radius of the system. So apparently the active turbulence is scale-free up to several millimeters. And I would just add with the fact that these structures that we observe are actually closely linked. So these large vortex that you can see here, if you wait long enough, will actually break down and form small vortices. And sometimes, but in more rare cases, the small vortices will coarsen and form these large vortices. So also here, there's still much more to be understood on a dynamical system, which is still very complex. And as an outlook is the last thing that we started to do is that we can actually go much further now with our 3D tracking algorithm. We can follow one bead that is transported through the turbulent state. And this is the trajectory of the bead. But we can not only record the trajectory, but you can also locally look around this bead how the bacteria are organized. So we can now have a look inside the turbulent state and link the transport properties with the local swimming properties of the bacteria. So this part, there's still a lot to be done. But a first step has been made in showing that actually this turbulence is scale-free. And with this, I want to thank you. And I want to show you the picture of our group from our yearly picnic in the summer on the Seine River. Thank you.
Thank you very much. Is there any question in the audience? You can write it in the chat. Match is saying bravo, I guess. Emilia too. Yeah, Emilia, you can ask your question.
Can you hear me? Yeah.
Yes.
Hey, thank you very much for the great talk. It's been amazing. Wow, so many beautiful things. I actually am super happy we had time to go to this last part with the higher concentration regimes, because indeed these patterns and the things that can happen at higher concentration. So my question would be in this part, if the system ever displays any kind of crystalline or glassy domains, and whether there's some, I don't know, macroscopic effects too, like interaction with light or something.
So there's many parts to this question and many possible answers. First of all, I wanted to say that concentrations, even our high concentration regimes, are still very small concentrations. So this is like 10%. Okay? So in our idea of a glassy systems where things really get into physical contact, we are not there yet. However, if we wanted to have collective motion at even more confined samples, if you go to higher concentrations, there's a different regime of collective motion where indeed steric interactions start to play a role. So in our case, we are not there yet because bacteria are actually still quite far. So nematic order is a good point. And actually there is, of course, some kind of nematic order because bacteria align to form these states, right? So you can measure order parameters. It's very difficult in the experiment, even if you started to do it, but from the theory, they can indeed measure that. And then you were wondering about interaction with light, right?
Yeah, just which would be a consequence of alignments at higher concentrations.
Oh, okay. You mean if they get birefringent or something like that?
Yeah.
So I don't know. Actually, they get also quite dense compared to the light. Because one of the things which makes the observation tricky, if you go to high samples and higher concentrations, is actually they get kind of dense. So I think it gets more and more difficult for the light to go through. And compared to a polymer that would stretch, I think they're rather big. It's like if you think about these colloidal liquid crystals, it's rather this kind of alignment I think that we would look at.
Okay, thank you. And also, yeah, one question was about what was the highest concentration, and these concentrations reported are in volume fraction, I imagine?
Yeah, it's volume fraction taking only the body into account. So of course this is debatable if you have fiber-like structures. Yeah, it's pure volume fraction taking the volume per body.
Okay, so not taking the head, you mean like?
Sorry, it's only the head, it's not the flagella.
Yeah. Okay, not the flagella. Okay.
Yeah.
Well, thank you very much.
Thank you.
Thank you, Emilia. Is there any other questions? Maybe in the meantime, I have one small question about the experimental devices, because you have different kind of experimental devices. Is it always the same? I don't know, I'm not working with bacteria, but same solution, let's say, to dilute your bacteria in each experimental device? Or is it like different? Like in the last one, is it the same solution or not?
Yeah.
And
So
does it play a role?
So if you think about the bacteria, there's different bacteria strains, which actually is true. I did not mention at all. So for example, we can have white type bacteria, which indeed run and tumble. We can have smooth swimmers, so we can suppress tumbling. So for example, this data was done with bacteria that do not tumble. And also depending on the different way we color things, is not always exactly the same bacteria strain. So this has small differences, and sometimes they can be crucial. And then the way that we present them is actually we put them in a minimal buffer. So we want them, so once we use them in the device, we want them to be happy to swim, but we don't want them to multiply. So it's a balance that if they are deprived of something, they will react with everything. So then they will go towards the oxygen, towards whatever. And so we want them to not interact with anything else. So we put them in a minimal buffer, which keeps them happy, but not happy enough to divide.
Okay, but you mentioned then that the last experimental device was oxygen transparent. So
yes,
if there is no oxygen, they are not moving, they are not doing anything?
Yeah, if there's no oxygen,
Okay.
so you can supplement them with something that can replace the oxygen, but normally they need oxygen. And this is why we work with PDMS devices, which if you have dilute bacteria in a microchannel is very easy, but if you start to have them in a big pool with a higher concentration, is something which gets limiting. Yeah.
Okay, it's just a question like that. Thank you.
Yeah, for sure.
Can I ask a question?
Sure, go ahead.
Okay. Hi, hello. Thank you a lot for your nice, very impressive presentation. I have a question. Maybe it's a little bit stupid, but I was wondering because, some bacteria are very known to be able to produce, for example, calcium. And in a setup with a bioreactor and a scaffold that has been printed, I was wondering if it's possible, maybe to introduce the flow of this kind of bacteria in order to promote bone regeneration or stuff like that. So I have just an idea like that. What do you think?
I know it's a nice idea. I don't know these bacteria so well that produce calcium. What I think one can do is that indeed using flows to direct bacteria to certain positions, I think is something very interesting. So you might want to avoid it if you don't want to have upstream contamination, but you also might want to trigger it. And indeed, here we have a lot of ways. So we know they orient upstream. We know they have a rheotactic drift towards one side. We know how they attach and detach from surfaces. So indeed, I think it's one of the paths that could be very interesting to design a geometry and a flow to bring them where you want to have them. Yeah, no, it's a very nice idea.
Okay. Oh, I keep it in mind.
Yeah, exactly.
Thank you, Mathilde. Michelle, do you have any last questions, or maybe in the chat? I didn't check in the chat.
No, at some point I had a question about these space correlations, but Anke answered it.
Okay, well done. I mean, you can always reach out also later if you have any comments or questions. So
very nice. Well, then I guess because it has been already a bit more than an hour that we're already talking. So yeah, if you have any further question, I'm sure that they can send you an email, or they can also comment in the YouTube underneath the YouTube video. And we can eventually send you the questions. Thank you very much. Thank you very much for the
My
talk.
pleasure.
I really appreciate it.